Welcome to On Air, a podcast from the AIR community. The community organizes and coordinates researchers studying all aspects of B and T cell receptor repertoires. The podcast is supported by the Antibody Society. For more information, please go to antibodysociety.org. This podcast has a special focus on clinical use of the adaptive immune receptor repertoires. We will look at how repertoires are currently used in the clinic and also discuss different opportunities where repertoires can be a great addition, the reasons why we are just not quite there yet, and how to overcome the obstacles. We are happy that you joined us for this episode of On Air. Welcome to the 9th episode of On Air, the podcast of the AIR community with special focus on clinical use of the adaptive immune receptor repertoires. Today with us we have Vadim Nazarov, co-founder and CEO of the startup ImmunoMind. Hello, Vadim.
Hello.
I am Ulrik Stervbo.
And I'm Cheng Ding. Hello, everybody, and welcome. So, Vadim, usually we like to get to know our guests a little better by understanding their background, specifically their journey on how they got to where they are, especially for yourself, right? You have this kind of alternative path as a scientist, and then also bringing it back to our AIR podcast, which is what brought you to work on the adaptive immune repertoire field and what excites you about it.
Thank you. Right. So my background is a mix between computer science and computer engineering, and I worked on optimizing algorithms and machine learning and developing machine learning algorithms. I worked at the network of laboratories led by Dr. Dmitry Chudakov. And I was a bioinformatician and later a senior bioinformatician there. I was leading projects on the intersection between computational immunology, adaptive immunity research, and machine learning. I really liked the idea of, as a machine learning guy, I really like the idea of adaptive immunity as a learning system. And I have my personal reasons going in immunology and working on something that is potentially has an amazing practical applications. This is very personal for me because my relatives and I were and is, let's say, affected by cancer and autoimmune diseases. And my path started with the analysis of the immune repertoire data for ankylosing spondylitis, also known as Bechtere's disease. Some of immune repertoire researchers know me by open source package TcR. I was a main developer of it, and I was a co-author of VDJ tools instrument, and later I became some sort of a team lead for immunarch, that is a successor to TcR. Again, it's open source R-package for data analysis of immune repertoire data. And my drive to do something practical from immune repertoire data has driven me to gather my own small research group focused on using machine learning in immune repertoires. So, for example, we created a machine learning model for classification for CD4, CD8 classification of TcRs using CDR3 and VJ sequences. I think it was many years ago, maybe around 2016 or something like that. I think the machine learning only started to become a very interesting topic in the immune repertoire space back then. And after that, I took a little bit of hiatus from science to recharge, to think through what I wanted to do. And I was invited to a position of director of AI and a co-founder to a Canadian startup focused on infrastructure for machine learning experiments. And after this startup, with my friends of, I think, 13 or 14 years, I usually call it a marriage, spending third of our lives, we started ImmunoMind with the idea of using AI to develop immunotherapies for cancer and autoimmune diseases.
So machine learning is what is tying all of these things together for you, coming from a strictly computational background into the field of biology and immunology, as I hear.
Of course, it's immune repertoires too, right? So consider me a person on the intersection between adaptive immunity, machine learning, and business side of things, because thanks to my work at this AI infrastructure startup, I learned more about the business side of things. And to be completely honest, I was, and I'm still very inspired by the business side of things, and that you can combine science and business to do something practical in a medical sense, right? So yeah, it's everything. And in addition to that, in ImmunoMind, we also found an amazing opportunity in single cell. So we are working on the intersection, immune repertoire research and single cell transcriptomics, site-seq, and so on data.
I have to admit, I'm kind of naive to ImmunoMind. So can you kind of give a background or an overview of what it is and what is the problem it's aiming to solve?
Sure. So we are focusing on developing more persistent cell therapies for cancer and autoimmune diseases. And persistence of cells is currently the biggest challenge in cell therapies. In blood cancers, in solid tumors, and autoimmune diseases as well. Cells simply don't survive enough to kill the tumor or to realize their potential. And what we do, we have a proprietary database of immune cells. We have our algorithms leveraging multi-omics data, immune repertoires, transcriptomics, site-seq, and so on, to develop more persistent cell therapies, to find new targets for these therapies, to develop leads, more persistent leads for found targets, and so on, going by the steps in the drug development pipeline. So in short, you can think about this as an AI-driven cell therapy company.
Is this more of a kind of a curated phenotype of a cell in a specific disease that you're trying to maintain? I guess I'm having a hard time wrapping my head around is how are you defining this persistent characteristic?
Right. Yes. It's a curated, well, it is a curated phenotype, because this persistence in reality it linked to many different properties of cells, right? To different phenotypes of cells. And you need to precisely calibrate your cell product to have the best combination of cell types, to have the best combination of cell types there. And you see the problem here, because cell products are very complex products. Those are biologics. So you not only need to develop it first in a very careful manner, right? On later stages, for example, in manufacturing, you also need to carefully monitor the composition of your cell product, right? So there are a lot of there is no one large task that is needed to be solved. This is a combination of smaller tasks because cell therapies treatment or biologics, they are different than small molecules.
Do you develop these therapies in-house, or do you provide the service for other people to develop? So with service, I mean, what to go for?
It's both, but our overall goal is to, of course, go towards towards developing cell therapies. because that's the reason I and my co-founder started this company, right? And this is our overarching goal. And it is the same model as with other AI for drug discovery or AI for drug development companies. They start as this hybrid service company slash drug development with universities, and later they build their own R&D facilities, and they acquire more and more data through the service model and through collaborations, and they became these drug development companies with a extremely large database of patient data and some very interesting AI technologies.
The database you need to do all this AI, where do you get the data for the beginning?
So, usually there are two parts: public data, and you can do something with this public data, or you can leave it be, right, and just compile together. And you can acquire data from partnerships, from collaborations, from alliances. We do both. We do both, and we did both. Because it's very important to have a proprietary, unique data that is only you have an access to from the business standpoint.
What are some of the major challenges that you face trying to realize ImmunoMind's goals and vision?
Well, there are typical risks for startups, right? And I'm not sure if I can figure out something new here. I think one of the most interesting things here, challenging and interesting, is of course the transformation from a scientist, from a bioinformatician who is doing immune repertoire data analysis and develops anti-developing packages, right, to a CEO of company that needs to hire new people. So, if you need to hire people, you need to build a framework for hiring, right? You need to do sales, you need to build a framework for sales, and so on, with fundraising and so on. So this change in not only in activities, but also in the mentality that you need to be productive, you need to be efficient, and you need to sadly you need to sacrifice the contemplating the beauty of the science here, because you need to get to results, right? And you need to get to minimally sensible results that you can demonstrate to stakeholders, you can demonstrate to customers, and so on. And I think it is still one of the biggest challenges for me. Well, another thing that is, I feel like, sometimes overlooked is the management skills. That when you go from managing 0 people to 1, and when you go from managing one person to 15 or to 20, it's really it feels like those are some leaps or some gaps that you really need to put your mind to think through, to overcome, and not just to let it go. Because if you want to be a good manager and good leader, you really need to reflect a lot on things and understand limitations of your mentality and previous culture. Because now you have a responsibility for 10, 15, 20 people, and you need to be efficient and productive.
Okay, so it sounds like a ever-evolving list of challenges that are never.
Yeah. So it's I'm not sure if it's some real story or a joke, but there is, let's call it a saying that you shouldn't ask an entrepreneur about how things are going, because they always respond that everything is bad, right? And yes, everything is always bad, but it doesn't mean there are no exciting parts, right? It's just because you always work on overcoming some challenges.
What about the technical challenges with sort of this immune repertoire and phenotyping and characterization, with it being more accepted or used for applications in pharma or for therapeutic benefits?
Well, the challenge that is always there, right? It is, of course, the quality of the data. And the question is, I think this is the key question in any AI application there, right? So how do you make sure that the quality of your data is good enough for algorithms or for any data insights you want to extract from the data. And I think about it from a different perspective than I usually see. That I think about it. What is the source of the data? And the source of the data is always in the communication between biologists, bioinformaticians, experimental people, right, and analysis people. I think it is some sort of as a conflict, as a hidden war between bioinformaticians and biologists, because bioinformaticians they don't see the full immunobiological picture, right, the full landscape of what's going on in terms of immunological processes. And biologists, sometimes they overlook some important parts in the for the data analysis. I don't know, additional time points, additional replicas, or so on, something like this, and. This is the reason we, for example, started to design experiments with our customers, with partners, with collaborators, because we know how to do a correct multi-omics experiment, and we don't want anyone to lose money and time on wrong experiments or on some So in order to fix this war, in order to be a middleman, right? We fix it by designing experiments with people, but still this is it's very interesting to see this never-ending war everywhere, even in large communities or large, how do you call it? Not in laboratories, but I don't know, in consortiums or something like that.
Yeah, I definitely, I'm an experimental biologist, and I definitely benefit talking to my computational partners very early on.
Yes. Yeah. I was, to be honest, I think about it a lot. And I think when I was building the first open source package, TcR, it was the bottleneck I found. It was how many years ago? 8 or 7. So I saw that the data is, the bottleneck is in the data. So I developed a function that automatically parses the input format. So you don't need to specify what is the data format of your files, right? And it's such a simple function, but you won't believe me how many positive emails I got. That okay, thank you so much for exactly this function, because sorry, I can't. It's too hard for me to manage all these different formats and files and so on. Right, and I think I don't think that sometimes I see that okay, let's improve data analysis skills of biologists or of experimental people, right, and so on. But I don't think this is the way because those are two completely different domains. I think biologists should learn what I called data quality skills. They just, in order to improve their productivity, they just need to learn more about what data is, right? To understand that the data is not something magical or ethereal, right? That it is a quite specific bytes and bits of information with some interesting properties. So, for example, there could be anomalies, there could be full data and not so full data. The data may be uniform or not. So, for example, some of the samples may lack the number of the huge number of immune receptors, right? Or some samples have a uniform distribution of counts of receptors, right? It's very interesting, but bioinformaticians, they usually don't have this domain knowledge to understand what's going on. So it's better for biologists to do this very, very first, not a data analysis, it's some sort of data pre-analysis or data quality check, where they could understand this meaning of the data and meaning of the experimental data, right? And possible problems with it or issues.
What you're saying is that experimental biology should extend from just being happy that there is no water contamination in a PCR, for instance, to start looking more into what is the shape of the bands that they get out, the intensity or whatever the readout might be. Is this the right interpretation?
Well, something like this, right? But I want to make sure, right, that so they focus on the bio side of things, right? That okay, look, statistics and machine learning is extremely hard. So if you feel like it's not for you, it's fine, right? You don't need to learn different types of distributions or even formulas for these distributions, right? But I think it would be better if people could at least understand why are those distributions there, right? Like, what is the typical distribution of immune receptor counts in T-cell receptor repertoires and in B-cell receptor repertoires, right? Those are completely different. B-cell receptor repertoires are much more uniform. And T-cell receptor repertoires follow the what's called the ZIPS distribution, so the exponential distribution, right? And if they find any anomalies, they could think. You see, they could think about their sources of these anomalies. When bioinformaticians, they don't have enough domain knowledge to catch these anomalies, to understand that, okay, look, something is not right, right?
So I guess if it's like a kind of an example or anecdote is if the distribution is off, and the two possibilities is there was a technical error or potentially it's a biological reason that is expected, like if there's a clonal expansion, etc. Or a disease sample, like oncology, hematological oncology.
Mm-hmm, mm-hmm. Yeah, yes, it is. And biologists at least could ask themselves this question, right? Okay, look, did I really expect this oligoclonality in this sample? Well, yes, because this is a person with an infectious disease and we have a very large expanded clone there, right? And bioinformaticians, a bioinformatician, if they are not very familiar, right, with immune repertoires, and we all know that bioinformaticians are their time and their minds are scattered across different laboratories, right? They simply again don't have enough time to learn the immune repertoire domain. And let's be honest, immune repertoires are extremely hard. And what's interesting, it's very interesting to observe how people from other domains, for example, from single cell, from transcriptomics domain, try to learn the immune repertoire domain. And despite knowledge of biology, despite knowledge of what are different types of cells and so on, right? They are, in my opinion, they are a little bit confused, or maybe sometimes they are shocked by the methods in immune repertoires, because I feel like immune repertoire methodology is more looks like an ecological studies, rather than your typical next generation transcriptomics data analysis routine, right? So it's really a change of mind there.
Would you then say that if the two domains are too separate, that this will lead to missed opportunities in terms of clinical development?
Mm-hmm. Yeah. Oh, I absolutely agree. And that's why we connect these domains, right? So we have people focused on single cell, we have people focused on immune repertoires, but of course they are communicating and so on. And I really think that the future I'm focusing on cell therapies, right? So in the first place, I'm talking about cell therapies. The future is definitely on the intersection between single cell technologies for omics, not immunomics, right? For other omics, particularly transcriptomics, and immune repertoire research. There is a fantastic publication by Juno Therapeutics called Clonal Kinetics something, where they track CAR T cells using immune repertoires and see what are the phenotypes of cells that are persistent, right? That are which are less exhausted, and what is the phenotype of cells which are less efficient in terms of anti-cancer therapeutics, right? This is, I think it's quite old publication from 2017 or maybe 2018. Sorry, I don't remember. But still, I think this is a very nice example of combining different fields, right? Because what is the an immune repertoire based medicine, right, or therapy, right? So when do you need immune repertoires? You need immune repertoires when your disease is linked to a specific receptor or a network of receptors, right? So combine it with transcriptomics information, and that's how you get cell therapies, for example.
How do you ensure that an AI classifier is based on exactly the thing that we expect it to be? So now we've been talking a bit about data quality and how to achieve this, but how do you actually know that the classifier is doing what it's supposed to be doing? So I'm asking this because I recently saw some example where cows in a field were classified because of the field. So a cow on a mountaintop was totally misclassified. And how do you check for these things?
Right. That's a great question. It's a continuation of the question related to the data quality, right? And I think that's very interesting that people think about AI as killer robots or something like this, right? But what's the most important in AI, in my opinion, is the everything except models. So the data in the first place. So I think let me start let me start with, again, non-computer science factor that I think plays a huge role. It's about reality versus expectations. And it's more on the psychological side of things, right? Because if people don't believe this closed box that outputs some predictions, nobody knows why, then you see it would be quite hard to help them understand that, okay, look, it is what we needed, right? So, and because this is about reality versus expectations, it means that it is about the faith people put in AI, and it's about education of people, or understanding the limitations of machine learning, and of course, it's a lot about machine learning software design, documentation, and we know that sometimes bioinformatics tools have fantastic documentation, and sometimes it would be great to have more documentation for some bioinformatics tools, right? But if with machine learning, it's another layer of complexity, maybe several layers of complexity, because of all the different components which are playing part in the machine learning process: input data, output data, model, train test set, and so on. So, of course, the ideal method to validate the output is the experimental validation, right? That you need to grab these cells that you output and check them in some mice model that, okay, they do kill tumor, or they do change some expression or something like that. So. Yeah, I'm not sure if I have more specific answer, because it's really it's about predicting the unknown. Right? It's about having a representative sample from a population, and well, how can we do it if we don't know what the population of immune receptors what it is, right? So. But you see, in immune repertoires, I think there are some fantastic achievements, despite my initial skepticism towards it, because I am a machine learning guy, right? I am extremely skeptical about any machine learning. Not very good for sales, because I'm always paranoid, right? But very good for results. So for example, we know that immune receptors they expand a lot in infectious diseases, right? So this is a fantastic approach to build foundation to build a machine learning model for this. For infectious disease. There is some interesting tools called, I think, Conga, T-Rex, and some others. I was skeptical first, but when I read publications on Congo, on T-Rex, well, I was quite impressed with the results. And you see, I am always extremely worried about using machine learning in immune repertoires, precisely because the potential space of immune receptors is too large, right? The number of possible sequences is too big, right? But no, it works. Those algorithms work, and I think they also had experimental validation. So okay. It's fantastic. So, in my opinion, it means that we really need to develop more algorithms, but for a very specific reason, to understand the limitations of this technology, right? Because, okay, in infectious diseases, it works. Maybe it won't work in cancer, right? Or in autoimmune diseases, because the nature of processes is so different that the model trained on the infectious disease dataset can't be transferred to another domain. So I am all hands for more and more experiments on machine learning in immune repertoires, but with experimental validation and with the goal of understanding the limits.
So you would say any model requires experimental validation. That is the way that you can truly assess if a cow on a mountaintop is actually recognized or not.
Yeah. So in order to make sure that your cow detector works fine, right? You need to take it to a completely different place with the same type or with the same species of cows, right? But probably with a different landscape to truly understand that everything is all right and we can count the cows for farmers or something like that.
Going back to kind of the data itself, and I guess continuing the cow analogy of, the diversity of images or sequences of TcR and BCR. Do you think you spoke about how ImmunoMind is generating their own proprietary repertoires, but there's also a lot of consortiums that are trying to publicly define the massive diversity of human repertoires. Where do you see sort of the field going? Is it more going to be like a proprietary private set of data information that's going to be distributed among a handful of companies, or ultimately a public data set, kind of like the NCBI for genetics?
Right. Thank you. Right. So a proprietary data from the business point of view, a proprietary data is a huge competitive advantage. Competitive advantage means that it would take a lot of time and money for someone to replicate this exact advantage. Well, here it's data, right? So if a startup uses a public data, it means, right, that anyone could use this exact public data. So it became more about the question of speed, that you will be able to hustle and to create a model or maybe find some new targets or something like that and patent it first. And it becomes a question of your own charisma and negotiations abilities, that you could explain to investors that pharma companies will never take this data and replicate your algorithms, right? And well, probably it won't, but what if it will, right? So I'm not sure. I'm not sure. I see this as a very major factor, right? That if we go with full public data, we remove many opportunities for businesses based on gathering data and selling it to others. And well, using this data, right, or gathering this data and using it for internal purposes. So it means that the government support or the grant support should be bigger here to compensate for the lack of these business-oriented agents, right? And it means that some active people who found themselves entrepreneurs, who found a soul of entrepreneur inside them, probably they would move to other domains because they don't see any opportunities here, right? But you see what's interesting. What's interesting here is that this is very interesting question. This is extremely interesting question, and it's very deep, because from the positive side, right? What if I'm an aspiring, I don't know, PhD student that gathered several students where we are machine learning specialists and we have some bright ideas, right? I think it's fantastic that this young student, young specialist, has an access to this public available data that they could use for anything, for experiments, for their diplomas, for getting to know the fantastic world of immune repertoires. Right. So, but one interesting downside of all of this picture is that data scale doesn't mean data quality, right? And but data quality, I mean like everything. I mean not only quality of the data, but quality of experiments. So, for example, additional time points, right, or replicas or something like that, and maybe more in-depth sequencing of data. So, for example, I remember quite big fail, I think, when we realized. I think I worked with some laboratory on a project or something like that, again, many years ago. We suddenly realized that we had to sort cells to CD4 and CD8. Well, and we didn't, right? But the whole idea was to find differences between immune sub-repertoires of CD8 and CD4, right? And now we have a completely useless experiment. Right. So the question is, who generates this data and, well, how? Because if you have smaller agents, smaller startups, smaller labs, they have this closed loop of motivation to generate. They generate high quality data for themselves. They build a therapy using they build a platform or a drug using this data, right? They get more money from pharma companies or building a new company, and they have more capabilities to generate even more high quality data, right? So this is a closed system, so to speak. But when we go to the world of larger systems, I think, it becomes quite hard, right? So you have many stakeholders. You need to clearly understand what is the data for, or maybe at least understand some opportunities of this data, right? And again, especially in immune repertoires. So So for example, I really think that probably we should look in the direction of combining single cell and immune repertoires. So is in combining transcriptomics and immune repertoire data together, because the amount of information you get from it is absolutely crazy, right? And with traditional immune repertoires, with bulk immune repertoires, I'm not sure whether you could easily combine it with a separate single cell transcriptomics if you want to.
One last question. So looking into the future, what area, diagnostics, therapeutics, cell therapies, or specific disease do you envision AIR sequencing to immediately benefit human health?
Mm-hmm. Right. Thank you for a tricky question. And it's also a little bit tricky for me because I don't like to be an expert. Because I like to be a person who have some strategic bets, right? So I definitely have a strategic bet on anything related to monitoring tumor progression or or markers of resistance of tumors, or maybe patient stratification, and there is also biomarkers for, I think, for leukemia. No, sorry, I don't remember. Again, it's another too many papers, right? There is biomarkers that measures which measure a diversity of tumor infiltrating receptors, right? And they use it for, again, patient certification and so on. So we already have this amazing application of immune repertoires in cancer, in monitoring, in biomarkers, right? So, but for the future, not for the now, but for the future, I do have a strategic bet on combining immune repertoires and single cell transcriptomics for cell therapies. And I think it's interesting. I think I saw a paper from adaptive immune repertoire community on using bulk immune receptors versus single cell immune receptors, and I think we don't agree on the potential of single cell immune receptors. But I'm very excited about combining different types of information, because in this case it's not 1 plus 1 equals 2, it's 1 times 1. Well, it not equals 1, right? It equals, I don't know, 10 here. So yeah, I'm not sure about infectious diseases. Well, there is, but I see, again, I mentioned this machine learning publication and so on, right? But I'm more excited about the cancer and autoimmune disease.
This brings us to the end of the 9th episode of On Air, the podcast of the AIR community, with special focus on clinical use of the adaptive immune receptor repertoires. The podcast is supported by the Antibody Society. Please go to the website antibodysociety.org to get more information about our sponsors. If you have any comments or questions, drop us a line at onair@aircommunity.org or tweet using the hashtag onair with 2 R's.
Well, thank you so much for talking with us, Vadim.
Thank you, Ching. Thank you so much for your tricky and complex questions.
Always. We will return in 1 month's time with more thoughts on the clinical use of AIR sequencing. The last seminar of this year of the iReceptor seminar series will be with Katharina and Keller at Frankfurt Cancer Institute in Germany. That's on November 24th. All links and contact information are in the show notes. This podcast is edited by Abdul Aziz of the comedy podcast Spout Lore. Thank you for listening to